Okay,
Thank
so
you
I'm
very
gonna,
much. Yeah,
you're
thank you
welcome.
very much, Jean-Loup. And you
I'm
can
gonna
start
share my screen.
sharing your screen.
Okay.
And we see it. Great. Thank you for giving Go to Cell seminar. And
Okay,
you can get
perfect.
started.
And let me close this window because it might, okay, good. All right. So first of all, thank you for the invitation uh, to present this seminar. And what I had in mind was to give you some information on what we have been looking at recently, and in particular to this question, can we compute with cellular metabolism? So it is not directly, we are not directly building a cell, but I hope that through the various work that we have done, you might find things that might be interesting and related to building a cell. So just a few words about myself and my research group. So we belong to the Michaelis Institute, which is a large microbiology laboratory in the Paris region. And we are affiliated with the National Institute of Agronomical and Environmental Research, the French Institute. And we're also part of the University of Paris-Saclay. So we have been working in the field of synthetic biology for quite some time. Myself, I started this in the US. I was working for the national labs, and I was part of the Joint Bioenergy Institute at the beginning of the work. And then I moved to France and started a group really dedicated to synthetic biology, um, with the particularity that this group is half computational, so a dry lab, and half molecular biology, so a wet lab. So we kind of combine the two together. So we have been working for now quite some time in engineering metabolic and genetic circuits, and we do this in vitro, primarily in cell-free systems. So I'll show some of this work, and I know that that is of interest to the build-a-cell community because I've seen some talks in the past in cell-free systems. But we also do this in vivo, and when we do it in vivo, it's with E. coli. So we have been historically working to do that for metabolic engineering. So just as a reminder, in metabolic engineering, you have a host organism that we call a chassis. And the goal is to find the enzymes that you need to add to the host organism in order to produce a target molecule. And doing so you are building metabolic pathways, and you can regulate this pathway with genetic circuits. So I'm not going to talk about this. What I will focus more on are the two other applications, which is first biosensor engineering, and I will present this just as a motivation to really go in the core of the presentation, which is going to be on biocomputing. And how does this relate to metabolic engineering? Let's say you want to sense a chemical, you want to strain that sense a chemical, a molecule. And so the way the strain can detect the molecule is and change its phenotype, because at the end of the day, that's what you want. You want to, for instance, produce a fluorescent protein or change the colorimetry of the strain. So therefore, change the genetic expression. So one way of doing this is that the molecule binds to a transcription factor or a riboregulator and change its activity, either activate the transcription factor or repress the transcription factor. And okay, so the issue is that there's not that many molecules that can directly activate or repress a transcription factor, and even less a riboswitch. The riboswitch is at the level of translation and transcription for transcription factors. But if you use all the knowledge that have been accumulated throughout the years in metabolic engineering, that is all these enzymes that we know that can transform molecules into other ones, you can actually build pathways that's going to degrade molecules that are not necessarily detectable into detectable one, which we call effector, those that will bind transcription factors or ribo switches. And this is essentially the technology that we have put in place for biosensor engineering. So now if you can detect several of these molecules, okay, or several signals through metabolic pathways, you can start to mix these metabolic pathways and, for instance, add signals, or subtract signals, and start to do some more sophisticated computation, which I will show later during the presentation. Okay, so that's to give you the framework of what we do. And to do this, we have developed throughout the years many software tools and protocols that about a year ago we put together into a framework that's called Galaxy. I don't know if you heard about the Galaxy workflow management system. It's a system that has been developed in the context of genomics, but that is now used in many, many different fields, mostly in system biology, but also in chemistry, and for various applications, cancer research, drug design, and a lot of work has been done in COVID more recently. And so the nice thing about Galaxy, it's maybe a little bit complicated to put in place, but once it's in place, every piece of software, or it can be also automated protocols and things like that, are in little boxes, okay? And you just have to chain, as a user, you have to change the boxes together in order to create workflows. So what you see here is a workflow to perform biosensor design and engineering. And basically what it does, it's an end-to-end process where you start by giving the molecule you wish to detect, then some transformation rules that are coming from the enzymes, all the enzymes basically that are known in large databases, and a list of detectable molecules. And what it will do, it basically will run a retrosynthesis software, which is also used in metabolic engineering, to find basically the pathways that will link these molecules to detect to the detectable one. And you can even go further, it's not shown here, but you can actually go down to actually designing plasmid or insertion vectors if you want to insert this into your strain. And even down further to get the oligos that you need to purchase or to get synthesized, to assemble them, depending on the assembly protocol. And finally, to write a script to a robot that will basically drive the robot. And we have, for instance, automatic script drivers for OpenTrons. So all of this has been a community effort. You have several people in France, but not only in the UK and in Spain, and also a company, Amiris, that participated to piling all these softwares and tools together. And they are now accessible in the tool shed, the Galaxy tool shed, that is maintained at Penn State University. And we have about 50 tools, and it's really a community effort, and tools are being added up, as we go. We started with about 12 tools, and now there are more than 50 tools. So anyway, with this, once you have done, you create these maps, and in this case, for instance, the detection of cocaine, cocaine can be detected via enzymatic transformation to inform benzoate, and benzoate is directly detectable by Benor, for instance, in E. coli. But you have other solutions, and you can click on these things, and get information of the enzymes and get information on the transcription factors and so on. And doing this by this technology that we call the sensing enabling metabolic pathway, you can really increase the number of detectable molecules. These can be drugs. Might be interesting to detect if drugs have been metabolized into your organism. It can be, of course, biomarkers or toxic compounds. This the way that works, in order to engineer, we basically do this usually in two modules. A module that contains the enzymes that will transform the non-detectable molecule into a detectable one, the effector, and then another module that contains the transcription factor and the reporter genes. And you can swap, the enzymes depending on the molecule you wish to detect and also the transcription factors. So here are examples of things that we have done in the past, more related to metabolic engineering. Malonyl-CoA is an important cofactor in many pathways. So here's an example of a biosensor that can detect various malonyl-CoA solutions using different strategies and different enzymes. We can also detect final compounds of pathways. These are pinocymbrin and aranine or flavanone. They are precursors of many flavonoids. It's a whole family of molecules. So it's interesting here that you detect the final molecules, but not an intermediate one like cinnamic acid. Here's another example where through an enzymatic transformation, you produce cyclose, which is a rare sugar. And here we screen a small library of enzymes, that perform this transformation and through a biosensor. So which one was the most effective? So these are now other examples more related to environment and medical issues like detection of nitroglycerin, okay? In E. coli, here's the detection of parathion. Parathion is one step away from the cyclosarin, which is the mustard gas. We could not do this in our lab. Was actually paid by the French army here, but it's just one step away, and you can see that you can nicely detect parathion, and it behaves the same way than the native effector, which is 4-nitrophenol. So basically, all the parathion is transformed in 4-nitrophenol, which is detected by this transcription factor, DMPR. And here's another example where you have two enzymes, that are transforming this chlorinated compound, which is a toxic compound found in rivers. Through true enzymatic transformation, you can generate this other compound that is detected by LINAR. So we have a whole panoply of these biosensors that we have built throughout the years. And we have also used these in a more clinical context, and in particular, we are interested by biomarkers that we extracted from HMDB. And we are interested by combining signals together. So we wanted the detections to reach a single effector molecule. And one molecule of interest is hydrogen peroxide because there are many reactions that produce hydrogen peroxide. So when you degrade a biomarker, you may end up producing hydrogen peroxide. As a matter of fact, you have more than 2,000 molecules in HMDB that can be degraded either in one step or two step into hydrogen peroxide. Hydrogen peroxide is detected by OxyR, and you can downstream of that express some GFP protein or any other fluorescent or colorimetric protein you wish. And what you see here is an example of detection of biomarker linked to prostate cancer. We had a grant to work on this. So sarcosine is probably the best known biomarker for prostate cancer, but choline is also another one, and lactate also. Lactate is not specific, but if you combine the three together, you get a more accurate, diagnostic of the disease. And we even tested this on clinical sample, on serum sample, and compared with some commercial kit, at least for lactate. And what you see here is that the response of the biosensor in blue roughly agree, with the commercial kit, which are the green dot. At least you differentiate the control from the positive samples. So here are two other examples. On urine now, urine sample, human urine sample. I'm going to focus more on the first one. The second one is more obvious for law enforcement reason, but the first one is an interesting one because epurate, monitoring the level of epurate in urine is a way of measuring the efficacy of a probiotic uh, using Nissel, E. coli Nissel, that is now really at the end of a clinical phase. And it's going to be probably commercially available. And the way that works is that the probiotic as an enzyme that degrades phenylalanine, and it's for the disease phenylketonuria, which is the inability of the patient to degrade phenylalanine. So the way that works is that the probiotic degrade phenylalanine into transinamate through an enzymatic transformation. And transinamate is naturally degraded by the liver of the patient into epuric acid. So the way to monitor if the treatment work is to just measure, the amount of hippuric acid you have in urine. However, nowadays, to do this, you need to use LCMS or HPLC. You can also do this with HPLC, but it's rather costly and lengthy, So if you have a biosensor, and this was actually a cell-free biosensor that we constructed here, it can be freeze-dry, and it can be also overlaid on paper. You can basically have a kit, that you can bring home to actually monitor and test if the treatment work just by looking at the level of hippurate that you have in your urine. Okay, so now that I've introduced and showed you some practical example on how these biosensors work, I'm going to move on to more sophisticated devices and circuits that are related to biocomputing. So we're going to start with looking at things in vitro, and then we're going to move in vivo. So in vitro is in cell-free, as I mentioned earlier. And in particular, we are going to look if we can engineer a perceptron and why we are doing this. So a perceptron first is the basic unit that you find in all artificial neural network. And a simple perceptron is just a list of input, and you make a weighted sum on these input, on these signals. And if you want a yes or no answer, you apply to this weighted sum an activation function, which for a 0-1 response is a sigmoid. And when you are dealing with more complex network, even deep learning network, you just have a stack of perceptron. It's just a repeat of the same idea. When you have only one perceptron, it's called a logistic regression. So typically nowadays, what people are doing to diagnose a disease, they are using a cohort, and they're going to measure, the concentrations of various biomarkers. These biomarkers could be proteins, they could be RNA, or they could be small molecules, metabolites. And once they have done this and they also have a control, so positive and negative, they train the perceptron or the deep neural network, if they have a deep neural network, in order to perform the diagnostic, to have a machine learning that can handle the diagnostic. So people have shown in the past, that it's a really good way of doing this for prostate cancer. And more recently, it's been extensively used for COVID-19, and in particular, this paper published in Cell in 2020, where they were able to distinguish patients just when they enter the hospital, and they were able to predict if they could develop a severe complication of COVID or just benign one and go back home. So all of this is great once you have trained your network. So what you do in order to diagnose a sample, you first need to quantify the biomarker. So if it's metabolite, it's going to be using metabolomics, protein proteomics, or RNA-seq for RNAs. And once you have these concentrations, you just feed the concentrations to this formula that has been trained, through the learning process, and it will give you a predicted, diagnostic, so a 0-1. So all of this is great, but it takes still some time and some money and expertise, to perform these metabolomics or these omics measurement. So what would be really great is that if we could instantiate, the trained network directly with some sort of a biological device, such that if the device would behave exactly like the trained network, then you would put the device in contact with a sample. And in the case of a positive, it would express a fluorescent protein. And in the case of a negative, it would express nothing. So you could directly perform the diagnostic and reply a 0-1 answer. So, but in order to do this, you're going to need to use back, what I just mentioned about these sensing enabling metabolic pathways. And you have to be able to transform your biomarkers through enzymatic transformation into an effector, an activator of a transcript factor. And once it's activated, you need to produce, let's say, a fluorescent protein. But it needs to behave like a perceptron, so that is making a weighted sum and having a sigmoid response, So for the expression of the fluorescent protein. Nature is helping us because, as we have seen already through many curves that have shown, the dose response curve, it pretty much follow an S-shaped curve. It's not a perfect sigmoid, but at least it's a sigmoid-like behavior. And the reason is that if you have too many activator, you are going to saturate your transcription factor, and you're not going to produce any more, fluorescent reporters. So this is the reason why it gets a saturation point, and you have this S shape. So for the enzymatic transformation now, what we need to do is also a weighted sum. And so in order to do this, if we look at the theory, and according to Michaelis-Menten, the theory of enzymatic transformation, if the concentrations of the biomarker you wish to detect is lower than the concentration of the enzyme that are going to transform your biomarker into an activator, which is generally the case because you are going to overexpress your enzymes, then the concentration of the product, so the activator here, is going to be the sum of the concentrations of the enzyme, some kinetic constant, and the concentration of the substrate, so which in this case is the biomarker. So clearly here, the weight is the product between this kinetic constant and the concentration of the enzyme. So it may be difficult to change the kinetic constants because you would have to do some enzyme engineering, but you can clearly change the enzyme expression level, in particular if you are in a cell-free system, because the only thing you have to do is to change the amount of DNA encoding for the enzymes. And as you can see here on a result that we obtained for the transformation of hippuric acid, which I already mentioned, into benzoic acid through the enzyme EPO, when you, let's take a fixed concentration, for instance, 200 micromolar of hippuric acid, when you increase the concentration of the EPO DNA, okay, you are going to increase your signal. You are going to increase your signal up to a certain point, which is about 3 nanomolar of EPO DNA, because at this stage, you produce so much DNA, so much EPO DNA, that you don't have enough resources to produce the GFP or whatever fluorescent protein you are using. And this is a well-known phenomena, where you have competition of resources in cell-free systems. But in any case, you can have a nice increase up to these 3 nanomolar, and you just need to cap, the maximum concentrations of EPO DNA to 3 nanomolar. And as you can see here, you can play with the weight of the signal, by decreasing from 3 nanomolar to 0. You can have a range of weights that you can play with. So with that in hand, in order to avoid to do this measurement for every possible enzymatic transformation, we developed a model which basically it's kind of a Hill model for the transcription and translation, and Michaelis-Menten model that we use for the enzymatic transformation. So we combine these things together, and as you can see, is that the model pretty much behave like the measurement. So we don't even have to do the measurement if we change the transfer factor, or if we change the enzymes that are involved into the systems. So now that we have everything in place, let's just look at one example and a targeted behavior. For instance, in this present case, we have 4 different molecules which we wish to detect. And so they are present or absent in our sample. So that's 16 cases total. And arbitrarily, let's say that the 3 first cases are negative cases and the rest are positives. And so first we need to find the enzymes, that are going to transform our molecules into a single effector, benzoate in this case. And for that, we use the retrosynthesis workflow in the Galaxy system that I showed earlier. So now we also need to find the weights, and this is a simple logistic regression. So you can find off the shelves, software that can do that for you once you define, the positives and the negatives. And here's a set of weights, that will basically tell you that when the sigmoid function is below 0.5, it's going to be negative, and when it's above 0.5, it's going to be positive. And these are the corresponding weight. So now we have the weights, And since we have a model that can give us the level of expressions of the DNA encoding for the enzymes corresponding to the weight, we just go back to our tables, to our model that I showed in the previous slides, and we know the amount of enzyme DNA that we need to put into our system. So we have now everything in place, and we can go on and do it. And what you can see is that the observed behavior pretty much follow the targeted behavior, so what we wanted. And the red dots, red circles that you see here are actually the predictions made by the model, which are also not that far from the observed behavior. So now you can even change, the perceptron, and you can change your training set. So now in this case, the same system, but only the first case is negative. It's basically an OR gate here. Okay, so the only thing you do is to change the weight. There are different weights in order to follow this behavior. And changing the weight is going to change the level of the amount of DNA you're going to put into your cell-free systems that it codes for the enzymes. And what you can see here again is that the observed behavior match quite well the targeted behavior. So all of this was done in cell-free, and still continuing on that, because biomarker of disease was shown only for small molecules here, metabolites, but biomarker of disease, and in particular, for instance, for prostate cancer, are not necessarily small molecules. They can be also RNA, non-coding RNA, for instance, or proteins. And in the case of prostate cancer, I already mentioned sarcosine, but PCA3 is also a non-coding RNA that is quite a good biomarker of prostate cancer. There's even a commercial kit just for PCA3. And LDH, lactate dehydrogenase, is also non-specific, but still a biomarker for prostate cancer. So the idea is, of course, by mixing the three together, you will increase the accuracy of your diagnostic. So in order to do this, we basically build individual sensors for each of these each of these molecules. Okay, so for sarcosine, so we transform sarcosine into hydrogen peroxide. We already mentioned this, detected by OxyR. So you have the dose response curve. For PCA3, we use a technology called the toll switch that was developed. It was a first paper published by Jim Collins' group on Zika virus, if I recall. And so we adapted this method for detecting PCA3, and you can see here the fluorescence level. And we did also the same for the detection of lactate dehydrogenase. So the way that works here, we want to dose, the amount of LDH. So the system works as follows. So you first start to add pyruvate into your system, and LDH is going to transform pyruvate into lactate, okay? And lactate is going to transform by LOX-X into hydrogen peroxide. And then we use the hydrogen peroxide detection mechanism that I showed earlier. So for a fixed amount of pyruvate, as you can see here, by increasing or decreasing the LDH amount, you're going to get different response. And you can see here a nice dose response curve where we increase the level, the amount of LDH that was put into the system. So similarly with the small molecules, we can also build a perceptron with this. And we have, so since it's a more complex system, we could develop a mechanistic model like the yield model with the Michaelis-Menten, but here there are many components into this. So we prefer to use an active learning procedure. So don't have time to go through this, but it's something that we developed throughout the years to increase, for instance, the productivity of cell-free systems, or also to optimize metabolic pathways. And there are several publications. And basically here we use this active learning procedure to find the weights, and find the amount of the various parts that, the amount as far as DNA is concerned, of the various parts that we put into our system so that it behaves like a perceptron. And so let's look at a simple targeted behavior where this is an AND gate, and this is the observed behavior that we obtain after running the cycle a few times. This is another example. So now it's a full OR gate, and here again, the observed behavior match the targeted behavior. And here you have a more complex system where you want to detect the metabolite and the enzyme, okay, or the RNA molecule. And so it needs still some improvement. So I had a PhD student working on this, and we are still finishing up a paper on that, but we are in a good shape to get this finished. So this is, by the way, as far as we know, the first multimodal, because we detect in the same system, an enzyme, a metabolite, and an RNA and a non-coding RNA. So all what I've shown so far, it's on cell-free systems. And of course, it would be great, if we could do the same, in wool cell and so therefore in vivo. But doing this in wool cell has some complication. First of all, in order to adjust the weight of the perceptron, which is really at the heart of the problem, it was fairly simple in cell-free because we just needed to modulate, the amount of DNA encoding for the various parts, the enzymes or the transcription factors or these things. So it's more difficult to do this in cells, because first your system might interfere, with the native metabolic pathways or native gene expressions and so on. And so you need to limit yourself to things that are orthogonal. And the other thing is that it's quite difficult, to fine-tune, the expression level, for instance, of an enzyme. So here you need to use, library of promoter, for instance, or change the RBS and so on. And so people have attempted to do this using quorum molecules, but they went really through quite difficult steps, in order to make the system work. So, but when we think about it, and just looking at the native, for instance, metabolic networks, or gene regulatory networks that you have in natural cells, these systems might be able, to also solve some complex problem without maybe doing much engineering. Just looking at the blob, this unicellular protist is able to find its way through a maze. And the way it does this is by producing some molecules, and detecting these molecules so that it doesn't go back to a place it's been before. Pseudomonas putida also has quite a sophisticated system, for quorum production and detection, which resemble, a small perceptron. And if you go to higher organisms, like plants, there are things that have been called the plant perceptrons. So you really have more sophisticated things there. And so perhaps, these organisms are capable of solving more problem than we think. And if we think about it, at the end of the day, organisms learn to survive, and maybe survival is one of the biggest problem. And so what we wanted to look further into this. So, and the way we phrased the problem was, let's say we have a strain, and we wish to probe if that strain is capable of solving a classification or a regression problem. So the way we are going to do this is that first we're going to take the problem, and a machine learning problem usually have features, that describe the element of the problem. And we are going first to transform these problem features into nutrient we are going to give to the strain. And using different nutrient, we create different phenotypes. And then we are going to read these phenotypes, for instance, metabolic production fluxes, and then transform again these through artificial neural network, into the readout of our problem. Okay, so this is the basic framework, but in order to train such a system, we need to be able to backpropagate a gradient that we have computing the error between the measurement, the prediction of the system and the actual readout, so the true and the predictions. And in order to do this, we need to backpropagate this gradient up to the first neural network that is going to transform the problem feature into nutrient we are going to give to the strain. But in order to backpropagate a gradient, we need to replace the strain by a model that will faithfully reproduce the behavior of the strain, at least as far as phenotype is concerned. So one of the most used model as far as metabolism is concerned is the steady state model. There's a whole community, developing these models for many strains. There are many, it's called genome scale metabolic models, and there are many models for many different species. And this is called flux balance analysis. So the way that works is that you basically solve a linear program, and the linear program, which tells we want to maximize some fluxes, and usually we take the growth rate, which is the rate of the biomass reaction. And this is subjected to constraint. And basically, the constraint is telling that you don't have accumulation of metabolites inside your cell. This is captured by a stoichiometric reaction, SV equals 0, where V are the fluxes of all the reactions that you have into your network. You also have some boundary conditions for all your fluxes, and notably an upper bound. And so this is great, and you can use this as a strain model, and it will give you the various phenotype depending on the nutrient composition you give into your growth medium. One of the problems with such a technique, while it's good at qualitative measurement, so that is, the system is very good at telling if for a given nutrient the strain will grow or not, it's more difficult to get good result as far as quantitation is concerned. For instance, here what you see are the regression coefficient that you have between measurement and predictions for just a growth rate. And what was done here is that E. coli DH5 alpha was grown with 110 actually different media composition by choosing between 1 to 4 nutrients that were added to M9. And what you can see here is that, well, the correlation coefficient is at best around 0.5, which is not that great. And the reason is that in the flux balance analysis model, like these genome scale models, the input is not really the nutrient concentration that you have in your growth medium, but the nutrient uptake fluxes inside the cell. And this is not known. So you can actually do measurement, if you have only glucose, but once you have combinations of nutrients, well, they all will depend on one another, and it's not directly, so you need to perform a lot of experiment in order to get these values. So perhaps one way of using the same idea of this prior and post network that I mentioned earlier is to actually build a system where we have a prior network that will map the nutrient concentration that we know when we do our experiment to these nutrient fluxes that are required, for a flux balance analysis. Another issue with the classical flux balance analysis is that the system does not allow, it's usually solved by a simplex algorithm, and the simplex algorithm is not compatible with gradient backpropagation. So we develop alternative techniques, which are borrowed from different fields. So the first one is borrowed from physics. So it's called physics-informed neural network. And basically, it's a surrogate of FBA, but that is gradient compatible. And as you can see here, with few iterations, you can reach a very good correlation coefficient. And you also respect the constraint of your problem, and in particular, this equation SV equals 0. So the second example is using upfield-like network, and this was developed by the math community, this solution. And you solve at the same time the linear program and the dual problems. Every linear program has a dual problem with variable u, and so it's faster, but it involves more matrix computations. And the last one is based on graph neural network. And it was borrowed by something that was actually, it was developed at the same time and we did, but it was developed for gene regulatory networks, and all these systems basically behave the same way. And they can, of course, be trained on experimental data. And this is what it's shown here, and make predictions that are at least more accurate than what you would get if you were going to use only the flux balance analysis alone. And so you reach, regression coefficient for predicted points. This is not during training here, of 0.78. We can even go further and train these systems not only on media composition, but also on gene knockout. And so there's quite nice data set that have been built around the biolog plate. And these biolog plates have different media composition in each of the wells. And what was done here in this study was, for each of the well, there was 120 different metabolic gene target that were knocked out. So at the end, you had 145 conditions, 120 knockouts, so that more than 17,000 data points. That's a very nice systems to work here. So this is the same idea, except that you have an additional requirement to your system that is, when during training, that you don't want the flux of the reaction of the genes that have been knocked out, to be greater than 0. So you put this into the so-called loss function. So what you see are both quantitative and qualitative results, of the hybrid model. So the regression coefficient is about 0.8. It's not written here. But in order to know if the strain will grow or not, this is a quantification. You get a area under the rock curve, of 0.90. If you are going to use the flux balance analysis alone, the results are not that good. Although you start to pick something, on the rock curve, and this is what I was mentioning earlier, that flux balance analysis is not that bad when you do qualitative predictions. So now let's go back to our problem of trying to solve some classification or regression problems. So here again, we start with problem features. We're going to transform these into nutrient, and we're going to read the phenotype of the strain. And from this phenotype, map that back to the readout of our problem. So in order to do this, we are going to replace the strain by these hybrid models, okay, that we call, by the way, artificial metabolic network. And with the various architectures that I showed earlier. So we start with a neural network, then we pass into this hybrid model, then the hybrid model produces a growth rate, and we move to the readout. So we first need to train the hybrid model. So we expanded on our initial 110 different media composition to 280. And as you can see, you get the same behavior. These are a correlation coefficient on predicted points, so 0.77. So it's fairly good. And so once the system is trained, okay, so then we can start to look at specific problems. Let's take, for instance, a problem that has nothing to do with E. coli. It's a problem that you can download, from a repository of people that are trying to learn machine learning. So this is a data set, where you try to predict the white wine quality. So it's a score between 0 and 10. So the way that works, we take the features of the problem, density, pH, sulfates, content, and so on, and we transform these into nutrient that we are going to give to the strain, okay, in this present case to the artificial metabolic network. Then we read the growth rate, and we map back this growth rate to the wine rating, And so you do this for each element of the training set, and this is what you get, during cross-validation on 3 different problems. So these are 3 regression problems: available CPU time, energy efficiency, and wine quality. And you see they have different features, And what's interesting is that, the strain and the AMN perform not as good as the state-of-the-art. And here we compare with the XGBoost, just trying to solve the problem directly. So it doesn't work as good, but it still follows the same thread. So it's lower, but when we have a good correlation coefficient, you still have both in the XGBoost and the AMN. So, of course, we are still continuing this work, and we're also doing, and this is my last slide, we are also doing this for more classical applications of these hybrid models, and these are funded applications for instance, trying to find the best gene deletion in Pseudomonas putidas to produce various molecules, uh, find the best gene deletion in Nithal in order to engineer a strain that will degrade two compounds that are involved in autism. Same thing, but diagnostic tool for COVID-19, like I mentioned, to classify benign from severe. And finally, to gene deletions to detect, to produce a compound that are detected by Raman spectrometer, and this is part of a European Union H2020 grant. And so I'm done. And so since I already acknowledged the funding, so I wanted to acknowledge the people that worked on this, and primarily the first 8 people here are PhD students that work on various aspects of what I've shown. So these are the most recent one, but these are more on the biosensor that I showed earlier. And I'm done. I'm gonna stop
Thank
sharing.
you.
I hope you're still here.
Thank you.
Okay. And
We are all here. Thank you so much. That was fantastic. There are some questions in chat. Can you see chat?
yes, so yeah, I can see, and okay, so I'm gonna read it and answer.
Yes.
And okay, so first, when you are training the network, where's the data coming from? For example, the prostate cancer marker is a training data set based on real data or simulated data. Um, okay, so both on prostate cancer and COVID-19, because we have done this, these are real data that were obtained on clinical sample and cohorts. Prostate cancer, we have not done it recently, but we are currently working on COVID. And so there is, for instance, a data set that has been published in Wuhan, the Cell paper 2020. And you have, concentrations of biomarker for severe and benign patient. And we have also our own cohort. I hope that answered so it's real data. Okay. Second questions, is your model diffusion limited or is diffusion not taken into account? Diffusion is not taken into account, depending on the model, that we are talking about. But actually, all the models that I mentioned a kinetic model for transcription translation and Michaelis Menten. So diffusion was not taken into account. And so that was for the cell-free systems. And flux balance analysis, no, diffusion is not taken into account. So these are for wool cell, and the issue of having models with wool cell, you could, with full kinetics parameter, but the problem is that we do not know the rate constant of the kinetic parameter. So this is why people are using more coarse grain model, based on this flux balance analysis, steady state assumption. But they have worked, at least, for finding the gene knockout strategies or things like that. They are not that bad, these models, And okay, so I get another, but it's just say great talk. Okay, so if you have other questions,
I,
be glad.
I, so I have a question that might be a little beyond your talk, but have you ever thought of using eukaryotic systems in that model, yeast or even mammalian cell-free?
Okay, so that
That would,
depends on what we are talking about. So in cell-free, we are involved in a project, but it's more to produce molecules, okay, rather than detect, I have to admit. But we are, yeah, indeed, using eukaryotic systems, mammalian systems, and also purified systems. So it is something that we have our hands on these other technology in cell-free. So now for the more the wool cell thing that I talked about, um, uh, since we are representing the strain, by a model, by a genome scale model. So I would say any strain that has a cognate model will do. Okay, we can process that. Okay, and so we have not done it because in my lab we work with E. coli primarily, but we could definitely use other strains. For instance, we are engaged in a project to work with Putida, but we could also do it on yeast and other strains. Yeah, of course. And and actually, what we'd like to do is to, at least as far as model is concerned, is to take different species and see and compare the ability of different species to solve the same set of problems, same set of regression problems, because they have different networks. We don't know yet what it's going to give us, but so it's more for fun, but we're gonna test that. That's for sure. And
I would, yeah,
as long as there is a
yeah,
model, we can do it, a good model. So this is also why we use these flux balance analysis things, because people have really developed that through many years. There are many models out there, but maybe someone published it and it's very ad hoc, or so. No, we wanted to have solid base, and we believe, that the model, at least that are in the big database, there's been some amount of work that's behind that. So this is why we use these.
yeah, makes sense. All right. Thank you so much. Thank you again for the fascinating talk, and have a
Okay.
great rest of you're done with Monday. So have a great evening.
Yeah, yeah, okay. Yeah, it's almost 7 p.m. Okay, okay. And have a good
All
afternoon
right.
for you. Okay, bye-bye. For
Bye.
the US, yeah. Bye-bye.